messenger secreted by cell in the endocrine glands to regulate body growth, metabolism, sexual development, and function. Properties of hormone. Most of the hormones are secreted directly into the bloodstream to be traveled in blood to the target cell or organ. Hormones are small organic molecules which is effective even in low concentration. However, hormone can act only on a certain target cell that have specific receptor for a particular hormone molecule. There are two main types of hormone, non-steroid hormone and the steroid hormone. There are three classes of hormone, amine, peptide or protein, and the steroid hormone. Amine is the hormone that derives from amino acid. The example of hormone is norepinephrine and noradrenaline. Peptide hormone are hormones that are water soluble. They make up the largest hormone group. Example is oxytocin and human growth hormone. The steroid hormones are lipid soluble and are derived from cholesterol. Example of steroid hormone is the male hormone testosterone that's secreted by the testes and the female hormone progesterone and estrogen. There are some differences between the type of hormones, steroid and non-steroid hormone. Steroid hormone is larger in size, while non-steroid hormone is smaller in size. Steroid hormone is lipid-soluble molecule and make it unable to permeate the cell membrane easily, while non-steroid hormone is water-soluble molecule and unable to permeate the cell membrane easily. Steroid hormones are synthesized from the cholesterol, while non-steroid hormones are from amino acid, peptide, or protein. Example of steroid hormone: cortisol, aldosterone, testosterone, progesterone, and estrogen. While example for the non-steroid hormone: adrenaline, glucagon, and insulin. Mechanism of hormone action. The endocrine system acts by releasing hormone that in turn trigger action in specific target cell. Receptor on target cell membrane bind only to one type of hormone. More than 50 human hormones have been identified. All act by binding to receptor molecule. The binding hormone changes the shape of the receptor, causing the response to the hormone. There are two mechanisms of hormone action on all target cells. Non-steroid hormone, which is the water-soluble, do not enter the cell but bind to the plasma membrane receptors, generating a chemical signal, which is the second messenger inside the target cell. Non-target cells, which lack the particular protein receptor, do not respond to the hormone. Since hormones are lipid-soluble or water-soluble, their mode of action or effect will be different. There are two mechanisms of hormone action on all target cells: gene activation and the cyclic AMP activation. Hormone action of gene activation. Lipid-soluble hormone diffuse out from blood capillary to the target cell. Lipid-soluble hormone then diffuse through the plasma membrane. Steroid hormone bind with the receptor in cytoplasm and forming a receptor hormone complex. Later, receptor hormone complex enter the nucleus and trigger gene transcription of a target gene to mRNA to be translated into protein that alter cell activity.
Steroid hormones are not water-soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
Hormone action of cyclic AMP activation. In this mechanism, the hormone acts as the first messenger and bind to the specific receptor protein in the plasma membrane to bring information to the plasma membrane of the target cell. CAMP acts as a secondary messenger which then activates other enzymes. It relays the message of the hormone inside the cell.
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of CAMP from ATP. The CAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.